In this video, we will discuss about impact of the whole body NPY1r knockout on the metastasis in the pancreatic cancer affected mice model. In the previous video, we have understood the impact of the NPR knockout only in the pancreas. But in this video, we will discuss the impact of NPY1r knockout in the whole mice model. So in this case also they check for like three different conditions. First one is which has this NPY1R present in the mice. Second one has a heterogeneous knockout where one of the allele of the NPY1R is knocked out, and in the third case is a homogeneous NPY1R knockout mice model. In that case, both the alleles of this NPY1R has been knocked out, and what they are able to see is that in terms of the overall survival is that it shows the similar trend what they have observed for this NPY1R pancreatic specific knockout. That is, there is not much statistically significant difference between all the three different conditions of the mice models that they have studied. However, when they check the liver metastasis, they see a statistically significant difference in both the cases, that is in the heterogeneous NPY1R knockout model and in the homogeneous NPY1 knockout models, and these results are also similar to the results that we have observed in our previous video, where this NPY1R knockout is only for the pancreatic cancers, only for the pancreatic tissues or pancreatic cells. And in the panel D, in the first row, we see a representative image of the liver where we observed the metastasis in the livers in the three different conditions, and we can see that in the wild type conditions the metastasis or the tumor cells are higher compared to the case where heterogeneous knockout has been seen. Liver of the heterogeneous knockout of NPY1R is when compared. So here in that case we see a less metastatic, less tumors are present in the livers where heterogeneous NPY1 knockout is there, and where homogeneous NPY1 knockout is mice models are been checked for this liver metastasis. They also see a lower metastasis compared to the case where NPY1R is present in the mice. And when we check the immunohistochemistry imaging, we can see that the immunohistochemistry imaging also shows the similar results, that is the metastasis is higher in the NPY1R wild type case compared to the heterogeneous NPY1R knockout in heterogeneous condition or in a homozygous conditions. And in order to find out the reason behind this, they also check the tumor compositions or tumor microenvironment compositions of the pancreatic cancer cells at the end point, and at that point they do not see any changes in any significant changes in the tumor microenvironment in the three different conditions. But when they check the when they check the gene expressions using the RNA sequencing at the tumor endpoints, they are able to see that some of the signaling pathways are downregulated and some of the signaling pathways are upregulated. And downregulating pathways are mainly somewhat related to the tumor suppressions, and upregulated pathways are related to the downregulated pathways are related to the tumor progressions. Yeah, that is the correct statement. And upregulated pathways are related to the tumor suppressions. In terms of the downregulated pathway, they see that the TGF-beta signaling pathways has been gone down, which is previously been reported as a tumor metastasis progressed cytokines, or we can say which triggers EMT, that is epithelial to mesenchymal transitions. And in terms of the upregulated pathway, most of the pathways are somewhat related to the tumor suppressions, and this gives us a hypothesis that it's not this NPY1R does not cause any changes into the surrounding environment, but it will this NPY1R present receptors leads to the intracellular changes in the cells in a way that will suppress the metastasis. I hope you understood, and those results are been shown in the supplemental figures. I hope you understood the whole body NPY1r knockout results and its similarities with the pancreatic cancer specific NPY1r knockout and the reason behind this. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.